Assalamu alaikum, my dear brothers and sisters. Yes, in this video particularly I am here to start with the 2 marks questions and answers in microbiology. Yes, my dear brothers and sisters, so don't skip this video. Let's start the video without wasting a single bit of time. See here, 2 marks questions play a vital role in your examination. Why? Because I am telling my dear brothers and sisters, if you score less marks, where if you score less marks in 10 mark question or 5 marks question, okay, but there is a chance of passing or scoring maximum marks in 2 marks. Why I am telling means, see here, every time in your examination 2 marks questions are fixed means they are just repeatedly asking the same questions. Okay, sometimes what they will do, they will collect the question from your 10 marks, 5 marks, sometimes directly 2 marks from the year of previous year question papers. Okay, so it's very easy to detect those questions, 2 marks questions. So see here, basically 2 marks questions are 10 in examination. As you know, 2 into 10 is equal to 20 marks. In this 20 marks, my dear brothers and sisters, you can easily score inshallah 14 marks you can easily score. Okay, 14 marks you can easily score for 20 marks. Now what's the pattern for getting that 14 marks? 14 marks is mandatory for you all, means you will get this, inshallah you will guess this 100% you will get, but above 14 means it's some complications, but that also you can get if you practice. But 14 is the mandatory marks you will get in examination by practicing these questions. Now for an example, see, you got how many marks? You got 10 marks in 10 marks questions, means you got one questions clicked there. Then in 5 marks questions you clicked there 20 marks. Then 20 plus 10 30. Here 14, how many? 34. You can easily pass your examination. 34, 44, it's a very good score, first class score. Okay, now that 2 marks questions only we will practice here with answers. It's very important with answer. So please subscribe my channel Enriched Pharmacy and watch the video until last and share with your friends. It is very important. Let's start the first question. Write contributions of Edward Jenner. See, first you have to write, basically I will tell you in 2 marks question how to write this 2 marks question. One trick I will tell you, you just write 3 to 4 lines. It's enough, maximum 3 to 4 lines. Okay, keep in this mind, 3 to 4 lines it's enough for your 2 marks questions. Remember this trick. Now first one, write the contributions of Edward Jenner. He discovered first vaccine for smallpox, then he discovered immunity to smallpox. Enough. The second question, what are contributions of Robert Koch? Okay, he discovered the anthrax cycle and bacteria responsible for tuberculosis and cholera. He also clarified that the disease is caused by the bacteria, means bacteria only causes the disease, was discovered by this Robert Koch. Koch postulate what you are telling, no, you can write that also, you can write that postulate also. Let's move towards the next topic, means next 2 marks questions. That is the third question. See, what is enriched media? The media which contains the nutrients to support the growth of microorganisms. Okay, such type of media is known as enriched media, and the examples are blood agar, chocolate agar, etc. Then differences between gram positive and gram negative cell wall. One more important point I want to tell you. See here, what 5 marks questions you are writing, no, the same questions are repeated where in 2 marks question. Maximum questions what you are completed in 5 marks, no, that completed questions, if they not ask in 5 marks means definitely they will ask in 2 marks. So what I am doing, no, I am already uploaded the important questions in microbiology, in pharmaceutical engineering, physical pharmaceutics, organic chemistry too. Just go and observe those videos. Why? Because means you can get the idea from which topic or from where the questions can be arises. You can easily get the idea by watching my videos. First go and watch those videos. Then difference between gram positive and gram negative. See, gram positive thin cell wall with a less lipopolysaccharide content. This lipopolysaccharide content is very important. Okay, it retains the crystal violet stain, appears purple in color. Which one? That is gram positive. Due to its thin cell wall, it having the less lipopolysaccharide content, what it will do, it will absorb the color, and it means it will retains crystal violet stain and appears purple in color. In gram negative, it is a thin cell wall, it does not retain color, appears pink in color, and it has a high amount of lipopolysaccharide content. You read that also. Clear? Let's move towards the fifth question. Limited questions are there, so watch my video until last. Limited questions are there, enough for your 2 marks. Then write the principle of sterility testing. Sterility testing principles here. Yes, the tests are based upon the principle that if microorganisms are placed in the medium which provides nutrients and water and kept a favorable temperature, the organisms will grow, and their presence can be indicated by turbidity in the original clear media. Means if the organism is present or not can be defined by the turbidity. Okay, in the broth culture, turbidity index. That is the principle of the sterility testing. Means we are testing the sterile nature of our preparation, of our discovery content. This is the fifth question. Now the sixth question, write bioindicators for thermal sterilization. It is also very important. Biological, not bioindicators or biological indicators or spore test or bioindicators. These are most expected by means of monitoring sterilization, because they assess the sterilization process by directly killing known as high resistant microorganisms. Means if high resistant microorganisms is present in our media or in our preparation, means bioindicator which were used for the thermal sterilization can be easily destroy those highly resistant microorganisms like geobacillus or bacillus species. Okay, even though they are having the high resistance, they can be killed by these bioindicators of thermal sterilization. Now what is primary cell culture? The cells which are directly taken from the tissue and set in culture media for development. Okay, means simple, those culture which is directly prepared, means for example this is the tissue. Okay, the tissue. Now what we are doing, we are directly taking the cells for the culture from this tissue. Okay, this is known as the simple or primary culture, primary cell culture. Example, endothelial cells, fibroblast examples. These all are examples for primary cell culture. Now the next one is what is a secondary cell culture? Primary or secondary, from these two one is fixed. These are subculture from primary cell culture. These undergo proliferation. Means these are subculture. Means they are taken from the primary only. What we are doing means just we are proliferating or just we are manipulating or multiplying this secondary cell culture. Secondary cell culture under the culture media by providing nutrients, water and essential requirements for this secondary cell culture. It is a subculture of which one? Primary cell culture. Now the next one is that is question number 9. Give 2 examples for alcoholic disinfectant. Simple. First you write the definition of this disinfectant. Okay, it is the chemicals which are used to inhibit or kill the growth of microorganisms. These are known as disinfectant. Example, example we have to give isopropyl alcohol, ethanol and phenol. These 3 are the example for the alcoholic disinfectant. Now the 10th question is classify clean area. What is this clean area? See here, this clean area, the question they ask from the aseptic rooms, means aseptic chapter, aseptic unit. Now see, clean area, an area with a defined particle and microbiological cleanliness standards. Means defined particles, some minute particles under, for example 0.5 microns or less than that. Okay, or 100 particles which are about 0.5 microns. Okay, means they should follow the standardity. Clean rooms are classified according to their number and the size of the particle permitted per volume of air. Means that particles, those particles which are permitted per volume of air according to the standards. This is the 10th question. Let's move toward the 11th question. What is this 11th question says? The 11th question says, what is a grade 100? See, class 100 or grade 100 defined same where there should be not more than 100 particles and so on respectively. Clean room has HEPA filters to clean the incoming area. Means the grade 100 means the particle should not cross the 100 particles. Means the area should not be more than 100 particles. There should not be more than 100 particles. Means some particles are there, no, which can be set by the standards. Those particles are good. Means good means they can be, if these particles are present, means your product can be supplied for the further preparations. Which below 100, that is a grade 100. Clean room has HEPA filters to clean the incoming area. Means that clean room, for an example, laminar airflow. These laminar airflow, they contain the HEPA filters. Okay, high efficiency filters. So what they are doing, they are just cleaning the air. Then the 12th question is, what is thermal death time? Thermal death time is how long it takes to kill the specific bacterium at a specific temperature. Means what the time required to kill the bacteria at the specific temperature. That's known as the thermal death time. For an example, endospore. What's the time required to kill these spores with the heat or thermal? That is known as thermal death time. Okay, next we will move towards the 13th question. What is D value? This D value also a very important question asked many times in our examination. See here, D value means the time required to kill 90% of the population of the particular microorganisms in a specific media at the specific temperature. Simple. Okay, how much time we have to require to kill how much? What the concentration? 90% of the microorganisms. That should be at a specific media and a specific temperature. Now the 14th question, what is a viable count? Viable count is the method used in the cell culture to determine number of living cells in a culture. We have to determine what the quantity of the cells are present in the culture. Okay, this is different from other cell counting techniques because it makes a difference between live and dead cell. There should be difference between live and dead cell, where in this viable count means we are separating, or we are distincting the live cells and the death cells. Clear? So this is the main advantage of this viable count, or the main occupation of this viable count. Now the last question, that is the 15th question, very important question. What are Rodac plates? See here, Rodac, they are abbreviation terms of replicate organism detection and counting. They are abbreviations of replicate organism detection and counting, can be used for microbiological control of all surfaces. For example, for textiles or other hand products, and the control of folding tables, conveyor belts, trolleys, containers. Means they are controlling all the surfaces. Which one? That is Rodac plates. So Rodac plates, it help to control all the surfaces, means surface media. Basically, for an example, see, this is the Rodac plate. We have placed the culture media in this. Now what these plates are doing, they will control all the surfaces. Okay, can be used here, replicate organism detection and counting can be used for microbiological control of all surfaces. They will control the surface, means they will control the microorganisms on the surface. This is the Rodac plates. Yes, my dear brothers and sisters, this is the important question which were repeatedly asked 2 marks in our examination, and these are enough with 5 marks and 10 marks. If these questions are not asked, means definitely they will select from the 5 to 10 marks. 100% I guaranteed that from these 15 questions in our examinations, 5 to 6 questions can be asked, inshallah. Definitely 5 to 6 questions. So please don't misuse this video, means you don't skip this video, you watch until last. And if you like this video, means subscribe my channel and share this video with your friends. Thank you. Assalamu alaikum.